Morning, everyone. Can you hear me okay? So thank you guys for joining us back this morning. We're going to start off the day by talking a little bit about what some of the initiatives that we're working on in the medical and scientific arena. So this is somewhat new. So Amy mentioned we had our first scientific roundtable yesterday. My son was diagnosed in 2020, and after that had the opportunity to start working with Amy to really think about, what are some areas that we could really be moving forward? What are some areas that we could try to drive the research? And so that's what I'm going to talk to you a little bit about today. So just a brief outline. I'm just going to do a quick introduction of myself. I want to share a little bit about what this MSAB means. What's the importance of this MSAB? Who are the key members right now? And I want all of you to be thinking during the context of this is really, which part do you want to play a role in? Because we need all of your help, because there's a lot to do. And we have just a couple of us right now that are really trying to move some of these things forward. So as part of having an MSAB is really defining the objectives. We'll go through each of those today and then closing with a call to action. So just briefly about myself. It's really sort of, I think my journey is a little bit interesting. I'm sure each of you have a really unique journey. One of my early sort of memories in my life is volunteering for Special Olympics. I did grad school in genetics, worked in a muscular dystrophy lab, ended up getting a PhD in genetics, and ultimately then spent my career really in rare disease drug discovery and development. Ultimately, I was leading rare diseases at ParkCell. It's a clinical trial research organization when our son was born and had a lot of health challenges, as you guys have all experienced. And interestingly, after being entered into the undiagnosed disease network, we had the genetics right in front of us, right? This is what I had been living and breathing my entire career in life. And now all of a sudden, it was my own son. And so we received a diagnosis in October of 2020. At that point in time, the question was, okay, well, this is something we need to just acknowledge. We actually left and went to Disney the next day just to take a minute. You kind of grieve the life that you thought you were going to have. And then you said, you know what, actually, I'm in genetics. My husband is in drug discovery and development. We have an obligation to do something here. We ended up finding out about N. Lauren Foundation. Oliver, my husband, found out about that just through LinkedIn and Instagram. And this is individuals who have N of 1 mutations. And there are drugs being developed for these people. We thought, oh, wait a second, our child is an N of 1. So reached out through LinkedIn, ended up talking to the CEO of this organization. And, after putting my parent hat on for a minute, kind of tossed that hat off. And I said, how can I help? I want to be a part of this. So now I'm with N. Lauren Foundation. So really the personal and professional is essentially one. I spend my day job working on drugs for individuals with unique mutations like Ethan and including Ethan. And so that's really what I think is one of the important pieces here is just I think a lot of the families that we've talked to, when we say, well, we're trying to think about a treatment, it's like, is that even a thing? Is that possible? It is possible. It's possible. We know it's happening for other genetic disorders. We have the technology now to be able to target mutations. And that's really that's sort of what my North Star and what the goal is here in the long term is that, feel this obligation and a privilege, honestly, very grateful for the education. I feel it's an obligation to be able to help our kiddos. So we have the opportunity to form the Medical Scientific Advisory Board about a year ago now. And so through the NORD organization, there are some specific requirements for NORD. And so we're registered with NORD to be able to kind of get some of that support as well. And this advisory committee is really driven towards having and bringing in the world experts in DYRK1A, bringing them together, connecting them. And I think what's really interesting is a lot of them don't actually have the connection with one another already. So now we're saying, oh, hey, Craig, you're in Australia. How about this researcher in France? She's doing a lot of what you're doing, and she has samples, and now all of a sudden they can connect too. So not only does it benefit us, but it also is going to benefit them, which will translate into more knowledge about DYRK1A. So this shows you the members of the MSAB at this point. Of course, Amy as our lead with myself. Dr. Van Baan is a MD PhD. She's in the Netherlands, and this is a great example of one of the centers of excellence that we'll touch on in a minute. I think Amy mentioned it yesterday as well. She's actually been designated specifically by Eurordis, think right, as a defined center of excellence for DYRK1A syndrome. And that's a really unique designation. And what that means is that she brings in patients who can then be seen by every single specialist. They then connect with one another, and they can have the full story for every single patient that they address. And so that's really, I think, something, at least for us personally, we're missing in the US. I don't know if anybody has that. I see you nodding, but. Yes, okay, that's great to know. And that's one of our objectives, is like we need to find some of these key clinicians. And that's what Dr. Van Baan, I mean, I don't think she necessarily, has sought out all of these different specialists intentionally until she decided to pull them together in a way to say, okay, well, she works on all these neurodevelopmental disorders, and none of them are just neurology or just GI or just ophthalmology, but they all need to really come together. And I think they actually do that even with the therapists, which is really a unique opportunity. Craig McIntosh is a postdoc fellow. He's at the University of Murdoch. He also gave a talk yesterday during the scientific roundtable. So if you're interested, we actually found out about Craig because, just one of those networking things during the pandemic. An old colleague of ours was like, hey, you know what? We have some interest in DYRK1A. We actually sent some of Ethan's cells, and so they started doing some work actually on his cells as well. So really looking at kind of the level of DYRK1A and thinking about how could we possibly upregulate DYRK1A in a meaningful way. Dr. Branicky has, Amy has a really nice story about Dr. Branicky, who really helped her kind of move into this space as well. Dr. Piton is a molecular geneticist, so she does a lot of the work, and she also did a presentation yesterday. So I think those were all recorded, so I encourage you to take a look at those. I'm really trying to characterize more so the clinical manifestations, thinking a lot about it doesn't necessarily affect us as much the diagnosis, but trying to get to diagnoses earlier. Nadia is another DYRK1A parent. She has an exceptional skill set in healthcare communication. She can turn any message into something very sophisticated and very meaningful very quickly. And then myself, who I already introduced myself. So I'm currently at the N. Lorem Foundation, and we live in North Carolina. So just wanted to summarize briefly the key points from this scientific roundtable. So we did have the 3 speakers yesterday. Craig talked to us a little bit about these targeted therapies called antisense oligonucleotides. So the approach here would be to try to upregulate DYRK1A. So all of our kids, which is defined as being haploinsufficiency, which means if we have, normal developmental individuals have 100% of DYRK1A, the affected individuals have 50%, something around there. So the goal is there any way that we can use technology to be able to increase that DYRK1A? So he was focusing on some of this early data. I think what he showed, one of the key messages that I took away is, even though there's some promising and interesting data, it's hard. And I think I can share that from my own personal professional experience. Drug discovery and development is not straightforward, but it's really exciting that we have somebody that's even beginning that work for us. So we're really happy to hear what he had to share. Dr. Wilsley presented on a frog model. And so I don't know about anybody who was there, but I never got so excited about a frog model before, but she was so enthusiastic about these frog models, and she had beautiful pictures as well. So she really had, a lot of interesting data that she's generated about looking at the effects of DYRK1A on all of the different organs, which is really fascinating, and then linked it some to microtubules and hemophilia, and a lot of these could potentially have therapeutic implications as well. And she also touched on a screen of some existing drugs that she's going to be doing on some gut symptoms. So hopefully maybe next year we could hear her kind of touch on some of the results that she posed that she'll be looking at. And then Dr. Piton did talk a little bit. She has a population of 60 individuals with DYRK1A syndrome in France that she works very closely with, and really, characterizing their clinical manifestations, and a couple of other pieces around the genotype-phenotype. So that's very high level. I'm sure I didn't capture all of what they shared, but those are some of the key things that I took away. So we've kind of set 5, rather large objectives for the MSAB. I think there's so much that we could do. And so, I think the goal here really, this is the first time we're doing this, the first time I'm engaging with all of you, and just really excited, honestly, want to hear your feedback. Like, let's think critically about like, what else do you feel like we need to know? Because we can access anybody in the world. And I think the other really cool thing, we have Simon Searchlight and we have Combined Brain and RareX, like these umbrella organizations. I mean, they have like 50 or 60 or many more other disease groups that are just like us, that have many of them been around much longer than we have. And so they have a lot of knowledge and experience that we can leverage. So any questions we can ask, we can probably figure out a path to finding an answer. So the first one seems obvious, but we just need to understand more about the individuals affected by DYRK1A syndrome. Second one is related to drug development. The third is funding research. So this is something, and I don't think we're touching on it this year, but just as far as how are we raising money as an advocacy group, and then what are we going to do with that money to really translate into benefit for our community? The fourth being defining the centers of excellence, and then the last is on educating the community. And so you'll see kind of each of these, we have a variable amount of progress. So, the first one really thinking about just how are we going to get a better understanding of the population? So I think one thing that's very evident is there is a broad range of symptoms. There's a broad range of severity. The mutations are unique. And so that's what we touched on yesterday. I know our son's mutation, he's an N of 1. Most of them are only seen in potentially a couple individuals. We don't actually have a lot of data yet to say, okay, well, if you look across the gene, mutations in this region lead to this. Mutations in this region lead to this. So I think that's really something that could be very powerful, because ultimately what we need to be able to do is understand the consequence of the mutation to think about how can we actually mitigate the effects of the mutation. So if we don't understand the mutation, what it's doing, we can't do anything about it. So that's kind of, we have to take a step back first. So in order to do that, and also on the clinical data characterization, we have these exceptional partnerships with RareX, with Simon Searchlight, and with the Tiger study. And so I'm going to touch a little bit on the RareX data, since they sent us a deck, and then you're going to hear a presentation from Simon and also from Tiger. I have a couple of slides from Combined Brain, but Anna Falzer's also here, so she's their CSO, so I'm not going to call you out, but of course there's plenty of questions that can be answered much better by her as well. So we can touch a little bit on that as well. So I think on the genotype-phenotype data, as I mentioned, Dr. Pitone is, really one of the key leaders in this space. So that's why it's so exceptional to actually have her on our MSAB. And so I think even, really what we're trying to even explore is, could we have like a postdoc or a student or some in one of these clinical genetics labs to really try to dig deep into this. And a lot of it, honestly, is pulling data together. So that's one of the key things that I, feel very strongly about in my personal and professional life. There are so many different sources of data and lots of individuals collecting data in lots of different places. So I think Anna and others who shared their experience with research yesterday, there's a lot of surveys for RareX, there's a lot of questionnaires for Simons. And so what are we doing to actually pull all these things together? So that's something we definitely want to be thinking about as well. And one of those aspects, actually, and Amy brought it up, was just as far as even for all of us who have shared our mutations and our locations and everything with Amy, and we have kind of a list as the organization, we haven't actually consented to share that, for her to share that with me, for us to even put it into a blinded summary. So I think we need to think critically about like what constitutes data. Is it okay to share it? And ultimately, how and why are we going to share it? What are we going to do with it? It's really important as well. So just touch briefly, like I said, RareX is a really exceptional organization as well that is doing a lot of this data collection. And so far, this is a real partnership. And I think one of the interesting things is we were able to be one of the first, I think, 15 as part of this partnership with RareX to help pilot their platform. And the goal is to really be able to kind of collect the data to really use in clinical trials. So I think really trying to think about how long it takes to study new medicines. And so it's really a lot of enabling the use even for data as a placebo. I think can't actually draw that conclusion that that's possible yet, but in one's mind, you could envision if there were a clinical trial and you could use data instead of having half the patients go on a placebo versus a treatment, that would be really an exceptional reason to actually be able to collect that data. And so here's a link here in the slide too, and definitely encourage you to talk to Kelly, who's out there. So this is really interesting. Actually, it's pretty exceptional that we have 49 individuals. I was really impressed by this actually. So congratulations to all you guys who are working on this, because I know it's a lot of survey data. So really, as far as the total participants, 49. I think we have over 600 families or 600 individuals who have been identified. I think with the explosion of whole genome sequencing, this is just going to grow. So, you know, the percentage of participants of our total population is not necessarily going to grow if we don't continue to work on that recruitment. So that's really one of the things we want to continue to work on. So for each of you who are not part of it, if you have the opportunity to learn more about it and consider it, and also influence others in the community, that would be great. So just to look a little bit more, breaking down, most of the individuals enrolled are in the US, but you can see the global breakdown here as well. Moving on into kind of what the actual results were showing. I think this is really starts to get very interesting, and I think it probably makes very much sense to most of us. Obviously, when you start to look at what are the key issues and which domains, the CNS is number one, digestive, behavior, vision, head, face, neck, growth, muscles. Obviously, you can kind of go through really trying to learn about what are the key areas that are the most important and what are the strongest issues that each individual has. And then when you look into kind of the CNS symptoms, really think about the cognitive impairment and seizures being the number 1 and 2. What the data centers are actually showing here, and going down into coordination, hypertonia, the abnormal EEG, which is often associated with seizures, hypotonia, migraines, and then down to cerebral palsy. If you look at the digestive system, what was really being shown here is around the constipation, feeding difficulties, issues with the esophagus, soiling defecation issues, stomach issues, and dysmotility. I think this really just starts to give a little bit more of a granular sense of what across the board, at least out of these 49 individuals who had the chance to enter the data, what they're really seeing here. So as far as the behavior and the psychiatric domain, the autism spectrum disorder was out of the 49, 70% did report this, as well as in the eye gaze, sort of the stereotypic behavior, repetitive behaviors, hyperactivity, the short attention span, and on. I don't need to read that to you, and you guys can have all these slides. But I think this really does start to show kind of some of the value and some of the things that we can learn. And also just thinking about the context, I think for us too, is like, so just getting a sense of like, these are some of the common elements that we really are all dealing with. And then we can translate this then into areas that we want to focus on for the clinical care as well. So here in the eye and vision domain, I think in the sort of eye movement issues for business, things like that, the farsightedness and optic nerve issues. I know at least my son was diagnosed with optic nerve hypoplasia as one of the many diagnoses they threw in and threw out pretty quickly. Nearsightedness and other visual issues. I mean, and this also poses an interesting, I think when you break it down by different systems, is really thinking about in the context of what is the most important, what is the most bothersome for each individual and for their caregivers, is really thinking about when vision is an important component. And that's something that oftentimes you do need to think about treating in a different way. So, and then on the head, face, and neck, I think the dysphagia obviously is a pretty major issue, oftentimes in the very young age. The inside of the mouth, abnormal nose, abnormal inside of the mouth and neck. I'm not entirely sure what those mean, so maybe we could follow up with Kelly to get a little bit more details on those. But if you guys have any questions on any of this, definitely feel free to reach out to Kelly, or if you're interested in learning more about the platform, the value, or just even considering to enroll, definitely let her know. So let's just touch on the genotype phenotype. So if we start to look at, again, without looking at the data from our own community, if we look in these sort of public databases, this is one of the more common databases that's often looked at called Nomad, that you can see just very clearly. And when you actually hover over these, you can see which mutation is which one. And you can really be able to kind of get a sense. Over 400 have been reported in these clinical databases. But if we know that we have over 600 or so individuals affected, then we know that we're not actually seeing the full population of individuals in the publicly reported databases. So that's an important piece of information for us to think about as we're trying to get a very broad and comprehensive picture. So Dr. Piton is working a lot on this, and I think we're going to continue to work with her on that. Oliver's doing a good amount of work on this too. And this is something, we really hope over time, if you could envision even on our website, so something very straightforward, just a picture of the gene and seeing the mutations across, if you can hover and see what are sort of the key characteristics, or, other ways that we can do some very straightforward visualization or sort of interactive kind of engagement to be able to understand what is the consequence of each mutation. And then really, again, thinking about what mutations have common characteristics, have common functional impact, and that we could really think about from a translational perspective collectively. Okay, so we can, if you guys have questions, feel free to stop at any point in time. Oh, go for it.
So you guys said that, of course, our kids have very different presentations of the symptoms, and you don't yet have enough data to identify which where the mutation has that. How much more data would you need? You have 49 families in this. If we got to 100, would that be enough? 150? Is there like a target where you could make an earlier diagnosis of how severe the condition will be?
Yeah, I'm not a clinician, so yeah, just for full disclaimer, I'm a geneticist. But I mean, I think it's a very good question. The strength of the data is only going to grow. So I don't think that you could necessarily say that you can't draw conclusions from 49, because you can draw some conclusions, and then you can caveat it with, well, and this is based on X number of people. I mean, because those 49 individuals, I don't actually know, but let's say they represented 49 different mutations. Then we do have some sense just from those individuals, but we won't be able to represent any other individuals beyond those. So I don't actually, it's a really, really good question, really interesting question. I don't have the answer to it. And I think the other thing is, so the rare X is not the only source of that. So I think rare X is going to provide a lot of color around that as well. But so let's just say we have 49 in rare X, and we have 60 in the Netherlands. We have then, another couple hundred at the group in France. I mean, so I think that's the thing. We can start to piece it together. And so that's what the goal here really is thinking about the fact that, like what you just said, is 49 is nice, but we have actually 600. So come on, myself included. So we're just trying to actually, engage as our family to try to get more data in as well. But I mean, I think we will start to draw some of those conclusions with the caveats. So I wouldn't say that we're going to say, oh, well, you know what, we can't draw any conclusions until we get to this threshold, because I think we may never get there. Who knows? So we need to just use the data that we have and then encourage more people to enter the data as much as we can. Yeah, it's a good question though.
So,
Yeah.
Sarah, is there, speaking of like 49 here, 60 there, is there any organization that is kind of spearheading getting all of that data together so they go, okay, mutation here, and then it's all in one place? Is there anybody doing that?
Yeah, it's just a really hard question, honestly. I mean, I think like Combined Brain, there are some of these other sort of umbrella groups, I think, do that, but they each collect their own. So they have their different sample sets. I can speak from NLORM, for example, if we're saying for a single patient, and we know that they have data from all these different data sets, we're having to work with Across Healthcare, which is a data platform. They have this matrix platform, and they have sort of the technical capabilities to pull data from all these different places. And so we haven't done that yet, but I think it's possible. So I think if you ask who that organization is, it's us,
Right.
ultimately.
Because they kind of go when you go, oh, you say there's, the number 45%
Yeah.
that have hypo, and 65% of the people are showing hypertony.
Mhm.
Well, like, is there some similarity in
Yeah.
where they're
Yeah.
having the For their
Yeah,
variations.
their mutations. Yeah, absolutely. No,
But
absolutely.
it would
And
be helpful to kind of go, oh, when it's here, we're saying more this.
no, exactly, right? That would be amazing. So I think that's the goal.
Okay.
So we'd love to hear and brainstorm and have more ideas. And yeah, I mean, Oliver's going to be working on a little bit of that, but I think we need more support with that too. So if you guys are interested, yeah, go ahead.
So we've had genetics done at the Children's Hospital in Philadelphia,
Mhm.
right? And then we've had it done at Nemours. And we've also had the whole genome sequencing, right? All of that. And every time they ask us if we can send for it to be public or go out into the world, we always say yes. Where is it going?
I know, it's just,
Like, if
it's
we
so.
don't have.
I know. Well, we can ask for it. So I think what I'm learning is, there are different ways you get into different data sets. So for example, and I don't want to speak out of turn if our Simon's colleague is here, or Anna, if you have anything you guys want to mention about this. But so I know, for example, the data doesn't just go somewhere. The data sits with them unless somebody requests to use it. So the idea, even with, if say we know that XYZ biotech company knows a lot about DYRK1A and they want to learn about this and they know that RareX is collecting data, they may go to RareX or they may go to Simon Searchlight. We've done that through NLORM to try to go to Simon Searchlight. So the nice thing about that is it's open access, but you have to have like an IRB approved protocol to be able to access it. So I think it's not, because it kind of feels like a red flag actually some of the time if you say it's just going everywhere, but it's actually, it means that it's accessible in a purposeful way, is at least my understanding on that.
What do you need more?
Nemours, yeah, I'm not familiar with Nemours at all, actually.
Yeah.
Okay, I'll have to follow up with you on that one.
They were the ones that identified race by facial recognition.
Oh, really?
For his blood work. and stuff like that.
Oh, really? Really interesting. Yeah. Okay.
So I guess just to add to these questions about all the different studies underway, last night's panel also talked about the importance of putting your information into each. Would you say that they all have their own individual merits, and so it does make sense to put our son's information into each and fill out the surveys for each?
Yeah, I would honestly, because, and I don't have enough data in my own mind to suggest otherwise. So I think they each have their own unique. So like Simon's and RareX are both sort of more of the longitudinal data with the clinical characteristics that I think is going to be accessible by researchers, and potentially pharma to try to learn and help us in the translation. Combined Brain is really in the clinical trial readiness. So, and they're collecting samples to try to think about if we needed, say, and Simon's as well, if we needed these different type of cell types to be accessible to, say, test drugs and things like that. Like that's where they each have kind of a unique element to them. And I do think that's a question, honestly, we've talked about a lot in our own family. It's just like what specifically. So I think the more we can work with those umbrella groups to help you guys, because I think I appreciate the question and it's just not crystal clear, honestly, what exactly how each one is completely different from one another, but I see value in each. So because they do feel like they're doing different things. But it are a lot of questionnaires and it will be a good amount of, repetitive work as well, so. Okay, so I think we touched a little bit on this before already, but just as far as the second goal, really thinking about therapeutics and the fact that we are in this exciting place in time. I mean, 10 something years ago, you couldn't even think about understanding the diagnosis as much, let alone trying to target it. So the more that we understand the consequences of the mutation, we do understand actually that like the majority of the DYRK1A mutations that cause DYRK1A syndrome are loss of function that lead to haploinsufficiency. So again, if we say, just for simplicity, our kiddos have 50% and we have 100% and we need to get them closer to 100. Interestingly, and I think what adds to a little bit of the complexity, there are some mutations that have been shown to behave in the opposite direction depending on their circumstance. So that's something that creates a little bit more complexity in the drug discovery and development. But the idea here is to create a toolkit. So let's just say, again, we have different, some organization who has this really great sort of engine to create a treatment that could help our our kids, but you know what's going to be delayed because we don't have cells for them to test it in. We don't have any in vitro models, or, you know, some of these other, the in vivo models, we don't have enough contact with the patients to be able to get a sense, or from the caregivers to get a sense of like, what would be important in a trial. So all of those things, and I think just being here with all of you, even this weekend, is we just start to plant these seeds. So if this were to ever come down the pipe, what would this look like for you if you wanted to engage in something that could potentially be meaningful to increase DYRK1A? Really think critically for yourself in your day life. Like, what would this actually look like? Those are the things we need to pull together now, because then when say a pharma company says, well, we're going to put together a protocol for a trial, we'll tell them what it should look like, right? We'll tell them exactly how this could go. And I think that's really the FDA is on board with this patient-focused drug development. So that's kind of what the overarching objective is there. And then mention kind of the different therapeutic strategies. The ASOs are very well understood. They're understood to be safe. We also know, I don't know if you all are familiar with nusinersen. It's a drug for spinal muscular atrophy. It's a classic example of these types of therapies. It's essentially gone from these kiddos who are born with a defect in their SMN gene, and they normally would pass by the time they're 1 or 2, and they're living completely full lives for antisense. And so this is an RNA targeted therapy. And so that's something what I think is on the leading edge for us at this point in time. Gene therapies, you probably hear a lot about gene therapies in the news. That would be like a one-off treatment. We're not there yet. I wouldn't put Ethan in the gene therapy trial, honestly, even if we had one right now. So I don't think we're quite there. But there are other gene editing, and we're definitely moving into this space. And so there's a lot of exciting, smart people thinking about how to try to manipulate and kind of support and change the consequence of these mutations. I think another piece that's important to mention is on positioning. So again, if you come back to funding and you think about wanting us to get attention for people to want to study DYRK1A. So our community is 600 individuals. How do you actually position a community of 600 individuals to a commercial entity? So there are a lot of other things that DYRK1A is involved in. And so obviously, even some of the most recent, I think there's a Simon's paper and probably many others that show DYRK1A involvement in autism in general. So, on the opposite end, 3 copies of DYRK1A is Down syndrome, part of the Down syndrome critical region. So there's a lot of other interesting elements of DYRK1A that we can really leverage to try to position DYRK1A to be more attractive, to get more attention, and others to really focus on trying to think about therapeutics as well. And the last piece we mentioned is on clinical trial readiness. And this is a couple of the slides that Anna had shared. And again, they have a table out, so I encourage you to chat with them. This is really a group of this consortium focused on the outcome measures and biomarkers across other neurodevelopmental disorders. And I think that's one of the things that I found the most advantageous. I mean, these communities are, they're just like us. Like, a lot of them have, a lot more advancements even in kind of their medical and scientific knowledge base and kind of their funding and the research that's going on. I think a lot of those things that are really helpful to learn from, and really this group really works to help, and Terry Jo is the head of this, and Terry Jo and Anna are both faculty at Vanderbilt. And so they really focus on trying to support and putting all of these pieces in place for these groups with neurodevelopmental disorders to really be able to kind of speed this path to clinical treatment. I think, I think one of the important pieces wanted to mention as well about Combined Brain is they continue to do a lot of exceptional work that's going to be moving across the whole spectrum. So this is from kind of the gene identification and helping really with that toolkit, back to kind of the in vivo, the rodent and cell models, this disease burden. We haven't done that yet, but I think they're going to have a student working on one of those for the DYRK1A syndrome too. So we'll be, hopefully we'll be able to present that at a future meeting. Really thinking about the patient-centered outcomes. So a lot of this comes back to kind of what the FDA is going to require. If we were to ever have a treatment, how do you show that something is actually beneficial and efficacious in a very small patient population? So really thinking about all of these things in their totality, and Combined Brain is really able to do that to help us. So Anna and her team are here because they're doing this roadshow, and so encourage you to really talk to them about some of the work that they're doing. Again, having some of these samples is really important, and I can tell you even from NMORM, we oftentimes have like a year or more delay to try to get samples or cells from a patient when you try to go through someone's physician. It's pretty amazing actually how long it can take, because trying to get a sample is usually requires a protocol, and some physicians don't have a protocol. And so, and even after you have the protocol, they have to grow, all these kind of things. So we're trying to just think one step ahead. So if we know this is coming, let's not wait till it happens. Let's be ready to go in advance. And so this is one of the key elements. They have a really robust biorepository. And let's see, I think that was sort of, I don't know if there was anything else on that sample collection, but I encourage you to go talk to the Combined Brain team at the table. So I mentioned as far as the targeted therapeutics in development at this point in time, we do have both Craig and NMORM are working on this, and this, the goal here is to try to critically think about, are there ways to upregulate DYRK1A? I think importantly, when you think about targeted therapies, there are some targeted therapies that literally target one person's mutation, and that means each of us need our own treatment, like precision medicine on steroids. There's another sort of non-allele selective approach, if you will, which is what we could do for DYRK1A with the upregulation, because the idea is that each of us have one normal and then one pathogenic copy. So we don't worry about the pathogenic because we can't do anything about that right now. We're going to target the normal for everyone. So the goal would be to say, can we target the normal and then increase the DYRK1A sufficiently, and that could potentially be a therapeutic to be across any mutation. So any questions or comments? Yeah.
About it, I did want to ask about the therapeutics, because like, what does that look like practically? Like in my mind, like, okay, if we upregulate DYRK1A, part of its mechanism is to increase brain size, right? And make more connections in the brain. And when you have a fully formed skull, how would that work, right? And so I guess and this is probably so early, you don't have the answers, no one has the advice. I guess I'm just practically speaking, what would this look like? What could this potentially target, and what would it change in children if this was possible without, I don't know,
Yeah.
science is the one thing I always keep getting stuck on, like, well, how can you, the brain.
Yeah, it's set, right. So some of it's set.
Model and show
Exactly.
the brain grew, like, so our kids, their skulls are.
Right.
I don't know, just can't answer probably.
No, I mean, it's important. I mean, it's a really important question. I mean, there's going to be timing, right, for every person. So what would be the most? So obviously with every treatment, earlier is going to be better. We know that for sure. And so I think there are a couple of things here. So for example, there's been an Angelman study not too long ago. These are adults. I think most of them are, much in their, late teens, early 20s, treated with an antisense that I believe speech was the outcome. So started to say words who had never said words before. I think there would, so I think that comes back to even the work that we saw from Dr. Wilsley yesterday, like the gut. Like, could we do anything with the gut? Could we change anything? I don't know. I don't think we have the answer. We do know, though, you can't change anatomy most of the time. I think another interesting piece in my own work is there's this mutation in a gene called FUS. There's these very young individuals who have ALS, who have this very severe, very, very severe form of ALS. And so this one girl, as an example, had she was on hospice. She was 17. She had trach. She was on, she literally almost passed a couple of times. She started to be treated with this ASO, and she's now actually walking. She's come off the trach. And so what that tells us too is the brain is actually a little bit more plastic than people might think. So it's not to say that we can change the size of the skull. I don't think anybody would think that. But I think the other piece is we have no idea. So the question then becomes to you as a parent or a caregiver, or to any of us, is like, there are benefit risks for all of this. So this doesn't go to say that there aren't going to be risks here. So then the question is, what could the potential benefit be? And is it worth it to you to figure that out? And it will always be, of course, up to one's choice, but yeah, go ahead.
Yeah, I was just gonna follow up on that. I mean, it's not necessarily like we're trying to change brain size, although that's like a complication of what we see from loss of DYRK1A early on. As Sarah mentioned, the brain is very, they call it plastic, or you hear about plasticity, meaning that it can change. So even though you have maybe a set brain size, the neural pathways change. Your neurogenesis can happen pre to documented that. So I think the question is not necessarily like where you change the size, but can you change connections or both? And the brain is continuously developing, 5 and then eventually beyond. So, if you develop a therapy, for the kiddos that get diagnosed early on, they may have the window of opportunity for actually the greatest benefit. But at the end of the day, like, we don't know. Things haven't been tested, and that's why it's important to engage with research, even translational research and animal models, so we can better understand these concepts. Because at this point, there hasn't been enough done in DYRK1A to really understand, okay, if we actually correct for DYRK1A, at what point is that going to be beneficial or not? And I think from a lot of the work that, Helen's done in the frog models, or, whether it's done in the mouse model, we know that, the differences in, let's say, neurons, like how they fire, how the synapses are created, like we have that information because those studies have been done. The question now is that if we create a therapy, how do we change that sort of
And I think one other piece I would just add to that. So we have another gal who had this KIP1A. It's a very different disorder, but as far as, some of these effects of the disease have already progressed and we know we can't reverse them. But she had a lot of seizures as well. So there, you start to think about like, what is something, if you think about what's the most bothersome? What is the most bothersome? So think about what we know we can't change these kind of things, but what we would want to really think about as a community is like, if we had to have in the context of like proof of evidence or for efficacy, we need to decide together, what would really be incredibly impactful is 5 more words, or maybe this many less seizures, or maybe maintaining no seizures, or, so trying to think quantitatively, because that's what we need to turn this into. And even for that girl that I mentioned, I mean, she literally was having thousands of seizures a day. I mean, like just almost in a constant state of seizures, and she's decreased so much. And this is an N of 1 drug just for her. And I think the other piece that's important is that the potency of that drug was not outrageous. So it wasn't a drug that would necessarily go to a commercial program. This was a drug just for her. But the potency of just enough, just a little bit of a change in that target for her has made pretty significant clinical impact. And so I think that's the other piece. So I think unfortunately, what I think we're both saying is like, there's a lot of unknowns, but we do have enough information. I have enough information to stand here and look in the eye and feel confident that I would actually have been treated with one of these ASOs. Because I think I would love to see a couple more words, or, a couple, like a lot of other things. And so that's why the other piece I think people don't realize is like the impact and the power that we have. Like this is ours to design. So I think we really would want to be thinking about what exactly would we want to have change the most. And that's why we need to be this sort of like powerhouse of an advocacy group, so that if anybody ever wants to do anything with DYRK1A, discuss this. We can help you get there because we know exactly how it needs to be done. So. Did you have another question? Okay.
Well, it was, so Grayson, we've had so many MRIs, but he has a shortened optic nerve, but other than that, they always say his brain's fine, right? So it just made me think when you were like, well, your brain can't grow. And they've always said his brain is normal, but I'm guessing what they meant was it was a normal size, normal structure, normal, right? It didn't mean all those ways and everything were.
Yeah. Apparently ours is normal also. I don't know.
Right, and we've never had that.
Yeah.
So,
Yeah.
So I think we need to.
And now we need to talk about the centers of excellence and who are our physicians, right? Like who's telling us what we feel like we need to hear and what's missing. Yeah, go ahead.
Like mind blown at the thought of like, stupid. So this is like the first thing.
Oh, thank you.
But, like some practical speaking and sort of like, sorry,
No, it's
like
okay.
we're seeing here with some data. I wonder if there's like a visual, like I'm just thinking like going to doctors, we have to like explain what little they're playing to professionals.
Mhm.
And all we think about that with like specialists, but you know.
Yeah.
like,
You're the specialist, right? Yeah, yeah.
So I just wonder if there's like something, I don't even know what I'm saying or asking, but like a visual for us as parents and caregivers to like take a packet of like, this is what we know, like
Yeah.
from our speculums, you know?
That's something that I feel that we are working towards, like, to
Yeah.
have something that we can provide, and that's where they come in and, get the data and stuff, and have something that we can bring them to other doctors so they can understand. It's a goal that we're working towards, and this is where they actually help in, because the more data they have, the more information they'll have, the more, accuracy that they can share and provide to other providers and educate more people.
At DYRK1A.org, there is, or there are overviews, there is kind of a leaflet, or you can look at it and can hand out.
Okay.
It's not it's accessible to anybody, it's not like the Facebook page where you have to. But
And also Gene Reviews is a good one. Yeah. Were you gonna say that? Yeah, Gene Reviews is great. And it's also, Gene Reviews is written by Dr. Van Bon, I think is the main lead author. So that's like, Gene Reviews is like a NCBI. Do you want to comment more on that?
is that, the Human News is by doctors who have been established as researchers in the areas. There has to be an author who is published on DYRK1A to actually have a Gene review, and DYRK1 has one, and it's also required that it's updated every 5 years. And I just looked at this, was updated in 2021, and whenever a new paper is published, then that data is then integrated into the new Gene review. So I think by all this, like through Tiger, through Searchlight, through DYRK, as we push out more publications, they will ultimately all be summarized with Gene review, and Gene review is, you could Google it right now. And Gene review is one word, so Gene Reviews is one word, and you can print that and give it to your doctor. It's the good, most updated sort of summary data on different things. It's a good start for something that is, and it has all the references. So if you're clinician wanting more detail about some specific information, go back.
Gene reviews, does it have a website, or how do you get to it?
So if you just Google Gene Review, DYRK1A, it will absolutely pop up. I imagine we can probably.
Yeah, we could send a couple of links too afterwards.
Got it.
So it's part of
NCBI.
Right.
Probably one thing that we can do, we can include a link from our DYRK1A.org website there. I think is one of the easy things that we can do, because then not only people here, but other people can easily find.
Yeah. Yeah, go ahead.
You had mentioned that, I don't remember the exact gene you were talking about, SMS or something.
SMN.
That they already are working on some type of therapeutic. What stands between us and reaching it? Is it sample size? Is it funding?
Yeah, it's a good question. That's a much larger population. So I think this, right now we're just looking at a few hundred people. So that comes back to that positioning. So we need to convince someone of the value and the necessity, because if and it's just business, but you have to have a commercial return for a drug company to invest many millions. I don't know. Oliver's at Pfizer, so he can.
Quick comment. I mean, we're talking about also diagnosis and how it's difficult to even get a diagnosis of DYRK1A. Oftentimes, you got to go through the whole exome sequencing to get it, and oftentimes geneticists aren't necessarily looking for it outright. I mean, spinal muscular atrophy is much easier to diagnose to know of. But I think like what we've seen even from DYRK1A continues to grow. There's more individuals every single year that are getting diagnosed. So my sense is that as we continue to grow our population and we continue to gather data, that's going to be a more attractive target for other companies that want to engage. So the more data that we have out, the more that we identify individuals earlier, or even individuals right now that maybe that haven't been diagnosed yet. And that's going to help just create the platform.
At one of the meetings, they said that research is going into the inhibition of the DYRK1A.
Yeah.
And Down syndrome, yeah.
Act with groups like that, where if they accidentally stumble on it, to stimulate it instead of,
Yeah.
suppress it, that we're gonna be able to get some of that data from them to figure out a way.
Yeah, I'm not personally enough, but I think that's part of, it's a really good point. And even Craig, like they're trying to think about how can we get funding for DYRK1A? Well, let's try to just focus on DYRK1A and get funding through Down syndrome. So, I mean, so I think people are thinking about it that way. But it's a good point. There's actually like a DYRK1 conference, or it might just be a DYRK conference even, like.
I had a chance to talk to, there's one of those companies trying to develop for Down syndrome in San Diego, really close to me. So I met the founder and CEO, talked quite lengthily. And the challenge that he mentioned, like we can absolutely collaborate, but to take the, like to get down the level of the protein is way easier than put up, that's our case.
Much
So he
easier.
mentioned in his work, it's like none of the work that he's doing, like the work itself, it would help us. Like he said, it would be like kind of, we start from scratch. We can collaborate in terms of funding and like kind of have more people interesting and like, but the research itself would not help us. At least is one company. I don't know all the others, but he said, unfortunately, not help.
Yeah, upregulation is hard. I think that's the thing. It is hard. It's not, and that's why most of the drugs that are out there, they'll be like a splicing, so they can change like sort of the genetic mechanisms and things like that. And the knockdown is very common. But it doesn't mean it can't be done, and we're gonna make it happen. So I know we're way over time, so I know you're standing. Because do you want me to just kind of breeze through the last?
Yeah,
So
just go ahead and finish.
Okay.
And then, Cora's next, so she might be
Okay.
able to answer some of the
Yeah,
questions. 'cause they
absolutely.
kinda-
Okay. So just briefly, we're just working on this, but the goal here is that we want to start raising money, of course, and you put a little plug in for the Run a K for DYRK1A that's coming up, so everybody get your running shoes ready to go and fundraising. So the goal right now, and the organization has been raising funds, and so really thinking about now that we actually have like a very sort of solidified strategy, if you will, for the growth of the organization and from the medical scientific perspective is like, what are we going to do to drive things? We're not just going to be responsive, we want to drive them. So this is an important one. Just sent this the other day, so we'll be hopefully launching this year. And even, we're not trying to fund for people to get millions of dollars of grants. We want to fund even, I've heard people say like, even just $5,000 to keep the lights on, that's something, right? We need to, so the goal here, and then next year, I think what we'll do is we'll be funding some research. We'll have those individuals come to our meetings so we can all hear from them. So each of you have the chance to ask, and then we can have this sort of positive feedback loop of getting more research that we feel is important for our community. So I know we don't have time to talk about this, and Cora may be talking about this more too, but I feel like we could have a follow-up call or something, because I'd love to get everyone's input on this. This just has a couple of different, different specialists. But back to the goal, I think we all feel like the cross-functional, the cross-specialist piece is so important for them to talk to one another and then translate that into what therapies, even PT and OT and all those things. I think without having those connections, it's really difficult. And then the last one is just on the educate and communicate. And I think, Sarah, you brought up a good point, and this is something the team is working on as far as like, what is a document to ensure that we have education for our community to give to others. I think we've heard recently, even like, and we're struggling in my son's school, it's like the other kids have no idea what this means, so are there even things we can do to help, even like a pamphlet to share with other kids that are young? Like, what is this? Why is the act so different? Some of those things that are also on our minds as well. Okay, so that's it. Let's just close with this. Thank you, guys. It's really a pleasure to meet all of you, and you're our people. So I'm really, really glad to kind of make this connection and look forward to a lot of follow-up conversations. And please volunteer if there's anything or everything you want to work on, even small things would be super helpful. So we'd love all of your help. And then also don't forget about signing up for all the research that's out there. So
Did you say when the run is?
yeah, it's our awareness day is August 21st. So it's usually a virtual.
The run's gonna be on August 20. It's Sunday before the awareness day. And the website, well, is in the draft mode because of like, I have to launch the t-shirt. We're gonna go back to orange as our shirt and make that weekend really orange in social media. But expect, until July 1st to be the official launch of the website and everything else.
Yep, and thank you so much and I just acknowledge like Amy, I don't think anybody's acknowledged Amy, I don't know where she is, but she always is just saying thank you to everyone else, but everybody that does anything is like just volunteering, and like Anna's doing this other website and she has like a full-time job, so just thank you to everyone, I look forward to